Welcome and thank you for being here. My name is Francesca Martinez, and today I'll be presenting a study published in Nature in 2019 by Chow et al. And the name of the study is Stunted Microbiota and Opportunistic Pathogen Colonization in Cesarean Section Birth. So, the background. So, prior studies showed birth mode affects infant gut microbiota. So, we know that. But most of them use 16S rRNA gene sequencing, which couldn't tell us exactly which strains were being passed on or where they came from. Also, few studies looked at the first days of life when microbial colonization is most dynamic. So this paper essentially fills in those gaps with high-resolution strain level data from a large cohort, providing the strongest evidence to date about disrupted maternal transmission and hospital-acquired colonization. Now, the main question. To simply put it, researchers wanted to know whether C-section delivery blocks the natural transfer of microbes from mother to baby, and whether this leaves the infant vulnerable to colonization by other potential harmful microbes from the hospital. So, do babies born via C-section miss out on important microbes from their mothers? Based on the paper that I read, I kind of broke it into three sections where it's we wanted to know, are babies missing out on important microbes? Are these babies instead colonized by bacteria from the hospital environment? And how do these early changes affect microbiome development over the first year? Hypothesis. C-section births result in altered microbiota composition due to reduced maternal strain transfer and increased colonization by environmental opportunists. So essentially, there is a difference between C-section and vaginal births. It could result in the sense of different microbiomes. It could result in reduced amount of a good microbiome and instead be more filled with environmental opportunist bacteria, which usually don't tend to be good. So the bottom line up front. How did the authors answer it? So they did an applied longitudinal metagenomic analysis to 1,679 gut microbiota samples taken at several time points during the neonatal period in infancy from 596 full-term babies born in the UK hospitals, along with 175 matched mothers. They compared the developmental of microbiota in infants born by C-section and vaginally. They monitor microbial composition, diversity, and maternal strain transmission using high-resolution whole genome shotgun sequencing and culture-based methods. Why should we care? I started diving into this study specifically not to put more on mothers. I am a mother myself. Actually, my mom had four daughters via C-section, and I had my child vaginally. So I find it kind of intriguing to study this. And I actually wanted to dive into it because in my idea is not to put more on mothers, like I said, but more like, okay, how can we give an advantage to all babies, no matter if how they're delivered, right? Or how they're fed, right? Like we want all babies to have the same advantage from the get-go. So I started getting into this study, but I wanted to share maybe why, as my audience, you guys should care. So nearly 30 to 50% of births in some countries are C-sections. So C-sections are on the rise. It does open the door for preventative interventions, which for me is why I was drawn to it. How can we provide the same advantages no matter how a baby is born? As well as with the number of C-sections rising worldwide, we need to consider how is this affecting not just birth outcomes, but lifelong health. So what's going on lifelong after this? If we understand the microbial disruptions happening, we can begin designing safe ways to correct them and maybe preventing disease years down the line. And a lot of the studies that I was reading as I was researching this is how early microbiota composition influences immune, metabolic, and cognitive development. And this is not based on the researchers, but there's a saying a lot in my family of like 3-3-3. The first 3 months of pregnancy is important. The 3 months of a baby's life and 3 years of a baby's life. So I find it kind of comical because it kind of relates to this biology aspect of it's so important from the very beginning, and our microbes play a huge role in the future. So the need. As I said, urgency. C-sections are rapidly rising globally. Um, so as well as disruptive microbial colonization is linked to long-term health risk, for example, like asthma, obesity, allergies, and health and clinical practices lack microbiome informed protocols. So like, yes, they do have a lot of um, SOPs, as you guys might know who work in the hospitals and, GMPs and all these other things on how to maintain these good clinical practices. But it needs to be further researched. How can we provide a really clean environment that can benefit a mother who is giving birth? And early intervention depends on understanding initial colonization patterns. The broader implications. So let's think about the broader implications as let's discuss about it as a class. Like, what do you guys think this could implicate in, you know, in other categories? In my opinion, I think it can implicate on the future health and how we could make a difference earlier on. And this could be also break down like a really good research for microbiology and realizing maybe there's other areas that we don't really study or don't get to study and could provide a lot of information. Methodology. So this study enrolled 596 healthy full-term infants from the UK hospital as part of the baby biome study. So fecal samples were collected during the neonatal period, day 4, day 7, and day 21, and later in infancy, 8.75 months. The research used whole genome shotgun sequencing to examine the microbiota in depth. For strain level insights, they cultured over 800 bacterial strains and sequenced their genomes. PermaNova statistical analysis determined which clinical factors, such as mode of delivery, most influence microbiota composition. So I kind of just highlighted on this slide what were the techniques being used, and then on the following slide, we'll get in a little bit into the definition of them because I personally don't have a lot of background on them. Techniques used: whole genome shotgun metagenomics, bacterial culturing and genome sequencing, PermaNova from microbiome variation, and strain, I really don't know how to say that one, for strain transmission. So let's look a little bit at the definition. Shotgun metagenomics is the sequencing of all the DNA in a fecal sample. And that helps us identify the microbial species and strains. Then we have PermaNova, which is a statistical test for my stats people out there, used to assess how clinical factors like delivery mode affect microbiome composition. We have the strain, the name I don't know how to pronounce, which is a tool to track maternal infant microbial strain transmission. It uses DNA variation. And then we have culturing and whole genome sequencing, Over 800 bacterial strains isolated and sequenced to examine resistance and virulence genes. So as you guys can tell, like we had to talk about, the background used 16S rRNA, and now this study goes into a lot of detail utilizing all these tools. All right, now to get to the good stuff. We have our first graph here, and it's looking at vaginal versus cesarean. And on our independent factor here in our x-axis, we have the sampling age groups, and then which is based on the days that we were speaking about earlier on. And then on our y, which is our dependent, is the relative mean abundance by percentage. So this is a bar chart, and what it's showing is the longitudinal changes in the mean relative abundance of bacterial genera in baby's gut microbiota during the neonatal period and infancy. So looking at some of the key results here, obviously you can see here like the red and what bacteria it's correlated with. And just right off the back, we see, oh, there's a lot of red here and there's a lot of blue here, right, guys? So, in the vaginally delivered babies, we can see that it's dominated by commensal genera. By that, what do I mean commensal? Commensal, we're talking about good bacteria or microbiome. That's how I want to for us to think about it in simple terms. And when I say opportunistic pathogens, just how it sounds, it's opportunistic. So, I mean, I'm not saying it's always bad, but sometimes when there's the opportunity, it flips a switch. Okay. So that's just how I want you guys to think about it. And now time to continue looking at this graph. So we can see that in vaginal, there is a lot of dominated by commensal genera, correct? And those are like Bifidobacterium, bacteroides, E. coli, and Parabacteroides. So these made up about, in combination, 68%. And then in C-section, we see it's depleted in commensal, so the opposite from the other side. And it's enriched in opportunistic pathogens like Enterococcus, Staphylococcus, Streptococcus, and Klebsiella and Enterobacter cloacae. Sorry, guys, if I'm butchering these names, but I'm trying my best. So these pathogens accounted for about 68% of the microbiota by day 4, as you guys can tell. So the takeaway of this graph is C-sections disrupt the normal colonization, favoring the hospital-associated pathogens over maternally transmitted commensal. Then we have the following figure. This is figure 3A from my paper. And what it's showing is your transmission frequency of maternal bacterial strains to babies during early neonatal and late infancy periods, comparing the vaginal and C-section once again. So in the vaginally delivered babies, we have high transmission, about 74.4% of maternal strains, especially bacteroides, parabacteroides, E. coli, and bifidobacterium. And then in the C-section babies, we have a significantly reduced transmission of 12.6%. Bacteroides. So bacterial transmission was rare in C-section babies and rarely caught up later. As you guys can look at the graph, we can see that. And the takeaway is that vaginal delivery facilitates early maternal microbial transfer, while C-section disrupts this critical window. And here you guys can see this is on the x-axis. Now we have the number of mother-baby pairs. And then now our y-axis is all of these bacterium, and it goes into detail of how much each was. And you can tell here in vaginal, and it goes into quite detail there. And then we have figure 3B from the article, which was actually next to each other, but I had to expand it. So what it shows here is the transmission events of B. vulgaris strain from mothers to babies over time. So I believe it was on that first 3A, which is boxed. And the key results is that vaginally delivered babies acquire maternal B. vulgaris strain early in neonatal period, while C-section babies showed almost no transmission even by infancy. So what's the takeaway? That bactericidal transmission is time sensitive and severely impaired by C-section. Now we have figure 3C, and what it's showing is a transmission of B. longum strains over time. So again, in 3A, you guys can see that these two are boxed, and now we're going into detail of what's going on. So both vaginally delivered and C-section babies acquired V. longum strains, but transmission occurred later in C-section babies. So the takeaway is like, unlike bacteriides, Bifidobacterium can colonize later, but timing differs by delivery mode. So my last figure that I wanted to get into detail was 4A. We can see all these pretty colors here. And now let's break down what they mean. What it's showing is the relatively abundance and the frequency of opportunistic pathogen in neonatal gut microbiota, comparing vaginal, again, and C-section. So on the left, we got some bad guys, and then now we're going to get into detail what's going on with these bad guys. Or not bad guys, but no, what we said, that if they have the opportunity, they could be bad guys. So on C-section, we see that 83.7% carried opportunistic pathogens versus the 49.4% in vaginal delivery. So pathogens like Enterococcus and all these other ones that we talked about are still abundant. These pathogens constituted about 30.4% of the microbiota. So mothers' low carriage of these pathogens suggests hospital environment as the source. And the takeaway of this is that C-section and hospital birth predispose infants to colonization by antimicrobial resistant pathogens. So now we need to consider the environment, and that's pretty much what this chart was highlighting and talking about in the article. And I thought it was pretty interesting. And then it also shows, yeah, it just shows all the possibilities of what happens in cesarean. And you can tell the blue line is a lot longer than the red line, especially on that first combined one. So you can see the obvious differences on the chart. And while the mother is always very low. So that's when it's like, okay, so the mother's actually really low, but now we're starting to see like vaginal and cesarean, no matter what, are still relatively there. Yeah, there's an obvious difference, but now we got to consider the environment. So, discussion. Practically, my study said, hey, babies delivered via C-section, it disrupts the microbiome, okay? And it can colonize the baby can be colonized by hospital opportunistic pathogens. And many of them with antibiotic resistance, and especially in a critical period for immune development. Some key interpretations is mode of delivery is a strong factor for neonatal gut microbiome. Lack of bacteriides, which are good, and presence of pathogens may impact. So immune development, metabolic programming, and long-term health. The findings support vaginal microbial transfer, like seeding trials, using of probiotics or maternal fecal transplants, and revisiting birth-related antibiotics policy. some of my conclusion. I know this seems like a lot. Also, I did want to say these images do not belong to me. They're just actually from Google Images, but I did not put it below the images. I just thought it would be cute to have some babies along the way. But anyways, so back to the topic. Mode of delivery has a strong lasting impact to neonatal gut microbiome. Vaginal birth promotes transmission of good microbiomes, and cesarean C-section disrupts it. And the hospital could also be related to this opportunistic pathogen. Many Many of these pathogens carry antimicrobial resistance and virulence genes. So other factors like maternal antibiotics and lack of breastfeeding can also alter microbiota development, and that's something the research also talked about. Sometimes, even though the baby is born vaginally, if the mother is taking certain certain antibiotics, it could affect. And also the lack of breastfeeding, which I remember one of my peers actually got into detail in her discussion. Sorry about that. Give me 1 second. So the findings suggest that early life microbiota disruptions could have long-term health consequences, sorry, that we had talked about earlier. And again, it talks about the birth environment, how important it is. And it's crazy to think about, like, as I was reading this, like years ago, how babies were born quite literally in people's houses, or I don't know, honestly, in their patio or during war. And so it was very interesting to see all of this. And I don't know, what do you guys think? Like, it's crazy to me. Like, I wonder how maybe back then it's exposure to so much bacteria, and yet babies seem to be healthy. Um, and I think that's definitely something that we can discuss as a class. Like, I found it very interesting that I'm like, oh, like hospitals are so clean and there's so many steps. I don't know. Like sometimes when I visit at a hospital and I realize how much nurses have and they're like, okay, glove up, like wash your hands, do this, do that. Cleaning up bodily fluids is a specific procedure. And I find that so intriguing that it's so clean, obviously, because we want to reduce these pathogenic bacteria and all these other bad things that could get onto our body. But like back in the day, some babies were born that way and everything. I wonder like what did their microbiome look? Obviously it might be a lot harder to look historically and see what happened. Um, but I find that to be an interesting topic for us. And I also find it pretty intriguing that this whole study navigated how delivery methods could affect the microbiome. Um, and I find that fascinating, and I wanted to share that with you guys. So, some questions for discussion is, should hospitals screen for opportunistic pathogens in C-section delivery rooms? I'm sure there's already procedures, but I do not think they're like constantly being checked every single time. But I don't know. Anyone that works at the hospital, feel free to share. For the second question, can breastfeeding or probiotics mitigate the effects of C-sections on microbiota? And for the third question, should vaginal seeding or maternal fecal microbiota transfer be considered for babies born via C-section? Um, Fourth one, could probiotics tailored for C-section babies restore bacteroides colonization, which are some good guys? And number five, how do antibiotics given during C-section confound these results? Um, so feel free at the end of this discussion to open up the floor for you guys and uh, what are your thoughts and what do you think about all of this? Um, I think there is a chance maybe probiotics could definitely help, or maybe vaginal seeding or maternal fecal microbiota transfer, since it's already done onto adults. So these are my references. I wanted to thank you guys so much for your attention and your time. And also, I am so sorry if you heard the fireworks in the background. I had to pause a couple times. I live in Orlando, so it can be crazy over here sometimes. Just wanted to thank you again and have a wonderful day.